My name is Elaine Vickers and this is my Cancer Basics video series. In this video, I'm going to explain how cells turn the information in their genes into proteins. Now you might think that this topic is a bit dry and uninteresting, but in order to understand how cancer treatments work, and particularly the newer, more targeted cancer treatments, you do need to know how cells use their DNA to make proteins. That's because targeted cancer treatments generally block a protein, but they only work if the patient's cancer cells contain a particular mutation in their DNA. In this video, I'm going to explain how DNA and proteins are connected. Let's start back here with the fact that cells contain chromosomes, chromosomes contain DNA, DNA contains genes, and genes are the instructions for making proteins. The problem, though, is that chromosomes live in the nucleus of the cell, whereas proteins are made in the main body of the cell cytoplasm. So there must be a go-between, something that takes the information from genes and transports it into the cytoplasm, and that go-between is called mRNA. In order to explain what mRNA is, I first want to delve a bit deeper into our cell's DNA. Now, if you zoom far enough in, way beyond anything that you can manage with any sort of microscope, you discover that DNA is made from two spiraling strands, and bridging the gap between those two strands are little chemicals called the DNA bases. There are 4 different bases: guanine, cytosine, adenine, and thymine. Each one has a unique chemical structure, but they're all pretty simple. Here's adenine. Chemists depict it like this, but it's a bit of a simplification because at each junction there's a carbon atom and there's also a couple of extra hydrogens. In total, there are 5 carbons, 5 hydrogens, and 5 nitrogens. The crucial bit that elevates the DNA bases from being simple chemical compounds to the foundation of all life on Earth is that due to the shape of the bases, A can only pair up with T, and C can only pair up with G. And the order in which the bases are lined up along the DNA strand, that's what we call the DNA sequence. To recap so far, DNA lives in the cell's nucleus. It's made from 2 spiraling strands that we call the double helix, and the DNA bases A, C, T, and G span the gap between the 2 strands. Okay, so now I want to explain how cells use their DNA in order to make proteins. The first step is for the cell to locate the gene it wants from within the 2 meters of DNA in its chromosomes. That, as you might imagine, is a whole story in itself, and one that I'm going to conveniently ignore. So here we have the gene that the cell has decided it wants to use to make a protein. It first separates the 2 strands of the gene's DNA, and then it brings in a protein that can make a copy of that gene, but the copy is slightly different from the original DNA. Firstly, the copy is only the length of the gene. Secondly, it's a single strand rather than a double helix. And thirdly, the T DNA base, thymine, has been swapped for a different one called uracil. The single strand copy of the gene is called mRNA. Now, it does get processed further. Again, I'm going to just ignore that for the time being. But that strand of mRNA containing the information from the gene, it leaves the nucleus, it enters the cell cytoplasm, and it finds a ribosome. That ribosome clamps onto the mRNA and then travels along it 3 bases at a time. Maybe it first finds a CCG, and then an AAU, and then a GUA. Depending on the order of the 3 bases, the ribosome plucks out of its surroundings the corresponding amino acid. It moves along, it adds another amino acid, and then another and another. There are 20 different amino acids. Again, they're relatively simple chemical compounds, but ones that can be linked together. And the ribosome, depending on the 3 bases it's just read, it will choose the amino acid that matches that set of 3 bases. And each set of 3 bases is called a codon. If the ribosome only links together a few amino acids, we'd call that a peptide. A longer chain of amino acids is what we call a protein. Now here we have a strand of mRNA and I've labelled each codon. Now I've added the name of the amino acids that each codon relates to. Amino acids are often abbreviated to a single letter, such as Y for tyrosine. Understanding this terminology means you can understand scientists when they talk about some of the mutations found in cancer cells. For example, around a third of human cancers contain mutations in a RAS gene, often the KRAS gene. Many of these mutations affect the 12th codon in the KRAS gene. This codon in the original DNA ought to be GGT, which corresponds to the amino acid glycine in the final protein. But if you change GGT to TGT, the ribosome will add a different amino acid, a cysteine. Scientists call this the KRAS G12C mutation. That's because the 12th amino acid in the KRAS protein is a C rather than the original healthy G. To recap, DNA is a complicated chemical made from two spiraling strands that we call the double helix. The strands are held together by the DNA bases. The order in which those DNA bases are lined up on the DNA strand is called the DNA sequence. If a cell wants to make a protein from a gene, it unravels the DNA for that gene, it makes a copy of it in the form of mRNA, and then the ribosomes use that mRNA as a code, telling them how to piece together the protein one amino acid at a time.